Welcome everybody, PGC members and others to the PGC Worldwide Lab meeting. I'm James Walters from Cardiff University and I'm chairing the session this afternoon with help from Tammy, and Pat sends his apologies, but we're excited for the first time to be able to involve early career researchers presenting at the lab meeting, and we're being quite ambitious in that respect, in that we've got 4 people lined up for this afternoon talking about ongoing or preliminary research or research that has been submitted using PGC and other data. So it should be an exciting time. If I can encourage people to submit questions as people are talking, then we can hopefully have a few minutes after each presenter to field those questions. We're going to stick quite strictly to the time so we get through the 4 speakers within the next hour. So thanks for cooperating with that. And without any further ado, I'm going to first of all introduce Renato Polimanti, who's from Yale. Renato, over to you. And if you want to share your screen, then you can get going. Thanks very much.
Okay. So now you should see my presentation. Okay, great. Okay. Thank you so much for let me presenting this data about major depression, alcohol dependence, and alcohol consumption, about the genetic overlap and the causality among these traits. This work is a collaboration between the depression and the substance use disorder working group, and I worked on this mainly with Eske Derske and Rosim Peterson. So as you well know, major depression and alcohol dependence are a common psychiatric disorder. They impact substantially the global morbidity, and they often co-occur. So epidemiological studies report that individuals with high MD are at increased risk for AD and the opposite. So we think that there are shared risk factors, both genetic and environmental, or may also be some causal process linking one disorder to another. From a genetic point of view, both twin studies that SNP-based analysis reported a shared genetic liability, and twin study reported a genetic correlation between 0.3 and 0.6. And individuals with elevated PRS for MD may also be at risk for AD. And a recent study also identified common risk loci as associated with comorbid AD-MD. So 2 potential mechanisms can explain this relationship: shared genetic mechanism or a causal relationship. Both these mechanisms are related to pleiotropy, which is the phenomenon where a single variant influences multiple phenotypes. And we can distinguish vertical pleiotropy and horizontal pleiotropy. With vertical pleiotropy, we have a single variant that affects one trait, and that first trait affects the second trait. With horizontal pleiotropy, we have the single variant affecting independently 2 independent traits. So we can use Mendelian randomization to investigate vertical pleiotropy. This method has some analogy with a randomized trial. That is a method that you can use to verify if there is a causal relationship between an exposure and an outcome. So a simple example can be the LDL cholesterol level. You can have 2 groups, they are matched for several confounders, and then you assign a treatment that lowers the LDL cholesterol, and then you follow up for a cardiovascular event. Now we can use genetic information to do a similar analysis, a Mendelian randomization. So we can distinguish 2 groups on the basis of their genetic information. So you have one group with genetically determined low LDL cholesterol, and then the other group, and follow up for the incidence of cardiovascular event. Obviously, to conduct a reliable Mendelian randomization, you have to respect some assumption. The first one is that your genetic instrument should be associated with the exposure, and the association should be really strong. And then the second assumption is that the genetic instrument should be unrelated to possible confounders that link your exposure and the outcome. And the third assumption is that the genetic instrument shouldn't be associated with the outcome other than through its association with the exposure. So these are the 3 main assumptions to conduct a Mendelian randomization. Obviously, there was an evolution of Mendelian randomization analysis. Is the two-sample Mendelian randomization. In a standard MR analysis, you have that a single sample have information about genetics, exposure, and outcome. With a two-sample Mendelian randomization, you can obtain information about the exposure and the outcome from two independent samples. This means that we can use GWAS summary statistics. There is no need to measure all the phenotypes in the same sample, and you can make causal inference through meta-analysis. Obviously, we can have potential biases in a two-sample Mendelian randomization, and is mainly due to horizontal pleiotropy. So if we link the genetic instrument to the exposure and the outcome independently, so we have to exclude this kind of effect. So there are multiple methods that people develop to conduct a two-sample Mendelian randomization. We can categorize them in three groups: mean-based, median-based, and mode-based. These different approaches have different power with respect to different scenarios, and they can adjust for some of the possible confounders. And within each of these methods, we can have various adjustments. So we can have fixed effect, random effects. We can apply a Radford framework. We can apply penalized procedures, and we can also apply a Stiger filtering. So there are multiple adjustments and multiple methods. And then when you run your Mendelian randomization analysis, you have to conduct sensitivity analysis to exclude the presence of possible bias. An initial analysis can be estimated the concordance across multiple methods, because you expect different power, but you should at least have the same effect direction. Then you can use an MR-Eager regression intercept to verify that your genetic instrument is associated with a count through horizontal pleiotropy. Then you can verify the presence of heterogeneity within the genetic instrument to verify the presence of possible outliers. And then you can perform a leave one out procedure where you test the same analysis, excluding one variant at a time from the genetic instrument to verify if the results that you are observing is driven by the contribution of a single variant. And then you can also run a funnel plot to graphically see if there are outliers in your analysis. So in our analysis, we use PGC, summary statistics for major depression, the one that was just a few weeks ago published on Nature Genetics by Brian and colleagues. We use the alcohol-dependent GWAS from the SUED groups from Walters and colleagues that is posted on bioRxiv. And then we also run two GWAS for alcohol consumption. One for alcohol frequency, that is a 6 classes scale from never to daily or almost daily. And the other one is the alcohol quantity consumption, is the average units per week. And these analyses were run on the UK Biobank. So first of all, we have to check the heritability and the genetic correlation among these traits. So we use the LDSC score regression. We have a significant reliability for all the 4 traits between 7% and 9%. And then we have a significant correlation. We have a significant correlation between AD and MD, about 42%. Is a positive correlation, and then we have a strong positive correlation between alcohol dependence and alcohol consumption quantity, that is around greater than 70%, and then we don't have a significant correlation between alcohol dependence and alcohol consumption frequency. And then we have a positive correlation between major depression and alcohol consumption quantity, and a negative correlation between major depression and alcohol consumption frequency, because alcohol consumption frequency is not an estimate of alcohol abuse. You can have a quote-unquote healthy drinking, one person that drink every day 1 glass of wine, and we know that the UK Biobank is wealthier and healthier than the UK general population. So this relationship, this negative correlation is not surprising. And then we obviously have a strong genetic correlation between alcohol consumption quantity and alcohol consumption frequency. So the MR analytic approach, this is what we use. We consider 2 genetic instruments, one based on the genome-wide significant loci and one on suggestive loci. So loci with a p less than 5 times per 10 to minus 5. We use multiple MR methods to account for different causal scenarios, and then we apply a Bonferroni correction accounting for the number of tests, 17, and the number of methods run for each test. And then we run several sensitivity analyses. So this is the results. Here I report the tests that have at least one Bonferroni significant result for one of the methods used. And we have 14 out of 17 that are Bonferroni significant. This is expected because we have a strong genetic correlation among all these traits. But then we have to run sensitivity analysis to distinguish between causality and horizontal pleiotropy. So the first thing is to check for concordance across all these methods. We can see that some of these tests passed the concordance analysis and some violated they didn't have concordance across all the methods. And then we have that almost all these results have strong significant heterogeneity. Only the MD-AD result passed this sensitivity analysis. And then we have the presence of horizontal pleiotropy. We can see that from the MRI intercept for most of the results. So we have only one result, the relationship between major depression and alcohol dependent that passed all these three main sensitivity analysis. And this is the details of these results. As you can see on the left in the table, we have the 28 different methods, and there is a concordant estimates across all these methods. As I said, each method has different power that is specific for a scenarios of causality. So we don't expect that all these methods give us significant results. We expect that they have similar estimate and mainly same effect direction. So we can see that for most of the results, we have at least a nominal significant results, and the estimate are generally similar. Then we can compare the MD result based on the genome-wide significant instrument and the one based on the suggestive genetic instrument. And we can see that, okay, the genome-wide significant genetic instrument is based on 40 variants. The suggestive one is based on 259 variants. We have basically the same estimate, 0.27 for the genome-wide significant instrument and 0.28 for the suggestive one. What improves is accuracy in the results. So is a stable results. As I said before, we don't have significant horizontal pleiotropy. The MRI gain intercept is not significant, and we don't have heterogeneity for this result. And this is the leave one out analysis. So is the same analysis excluding one variant at a time, and we have a stable result. So this means that no single variant is affecting the overall result. And then the final plot analysis, we have a nice symmetry, so there are no outliers that are affecting our results. So briefly, to show you what looks like one of the results that didn't pass the sensitivity analysis. So you can see on the left that methods have opposite effect direction, and only in 2 cases we have significant result. And if we look, the significant results are in opposite direction. So for the inverse weighted method, we have a positive relation 10 to minus 6. And for the Eager fixed effect, we have a negative relationship 10 to the minus 3. This means that the result is not stable. And then also when we compare the genome-wide significant genetic instrument and the suggestive genetic instrument, we have in both cases significant result, but in opposite direction. And this is for the same method. And we have a trend significance for the MR Eager intercept. It means that we have some evidence of horizontal pleiotropy and a very strong heterogeneity in this genetic instrument. So in summary, we have a causal effect of MD on alcohol dependence, which doesn't appear to be biased by confounders. The other significant results show non-consistencies across MR methods due to the presence of horizontal pleiotropy and heterogeneity. We cannot exclude the presence of causality, but we have strong evidence of horizontal pleiotropy, so we cannot say anything about these other results. And finally, for the relationship between AD on MD, we don't have any nominally or trend significance, but we cannot exclude that our analysis is not powerful enough. Indeed, the AD GWAS is relatively small. It's much smaller than UK Biobank, so we cannot exclude a bidirectionally causal effect. So this is the investigative team. As I said, I worked mainly with Roseanne Peterson and LDSC, but we had also many other collaborators from the SUD and the MDD work group from PGC.
Renato, thanks very much indeed. That's really Interesting work. So we've got one question from David Curtis who comments that in the first set of results that you presented, so those that didn't seem subject, I assume it's those that didn't seem subject to the horizontal pleiotropy or heterogeneity, he struggled to understand why the result was highly significant when the effect seemed smaller than the standard error. So perhaps you could go back to that slide and...
Okay. Here? Yeah, you are right. Probably the standard error is not the standard error. Is another value. Is not the right one. Yes.
Okay.
It's a typo.
Okay.
Because it probably is the 95% confidence interval, and is the upper result is a mistake.
Okay, so it's the upper threshold of the 95%.
Yes.
Fine. Good. Has anyone else got any questions they want to ask of Renato? Okay, in which case I think we should move on. Thanks very much, Renato. And I'm sure you'd be happy for people to approach you if they've got further questions.
Yes, sure.
Great. And thanks for your contribution. So next is Paola Giusti, who works in UNC with Pat Sullivan. So Paola, over to you. And I can see it's sharing your screen already. So
thank you.
We're running to time at the moment, which is good.
Yeah. And I'm going to set up my timer just so that I can keep track.
Great. And if anyone's got questions, then the best way is to go through the Q&A facility that's on Zoom, as David Curtis has done. Thanks.
Okay, so thank you for having me today. I'm going to be talking about our efforts to use chromatin interaction data to better understand genomic findings for schizophrenia GWAS. And so first, there isn't an easy exome path for schizophrenia. Exome studies for schizophrenia have not been as successful as they've been for other psychiatric disorders. On the other hand, GWAS has yielded many promising leads, but sort of we're still scratching the surface when it comes to the implication of these findings and connecting GWAS SNPs to genes and pathways. So as you all know, the genome is not a one-dimensional string. The 3D arrangement of chromosomes allows for the highly organized genome packing, such that genomic regions that are megabases apart or in different chromosomes can come together and interact. So here I'm showing a diagram. I'm gonna give a very brief overview of chromosome organization, so that on the left you have a diagram of chromosome territories. If you zoom into that, you can see this sub-megabase domains known as TADs, or topologically associated domains. And these are large genomic structures that are conserved across cell types and are highly conserved across species. If we zoom into these TADs, we can see these sub-TAD domains, and some of them are known as loops, and these come in different flavors, and they allow for organization and also of regulation of gene expression, etc. So we are interested in using chromatin interaction data to try to better connect schizophrenia loci to genes. The main approach that is used for this is we can use a number of techniques within the family of chromatin conformation capture approaches. Specifically, eHi-C allows for the genome-wide mapping of chromatin interactions. And for our purpose, we used a variation of eHi-C known as eHi-C that was developed by one of our collaborators, Fulay Jain at Case Western Reserve University. And the main thing that you need to know about eHi-C is that it uses a much lower input than regular eHi-C, and so it's amenable to postmortem tissues and samples of limited amounts. Just a quick overview, we did eHi-C on 3 fetal cerebra and 3 adult anterior temporal cortex samples. And we produced over 10 billion reads for these samples. We also incorporated all publicly available eHi-C data sets for human tissues. And so our main question was trying to figure out what genes and biological processes are implied by these connections. So the key readouts for the eHi-C data to keep in mind through the rest of the slides are fires, which are frequently interacting regions. These are an architectural feature of chromatin organization. Generally, they are enriched for super enhancers and also for GWAS SNPs. We looked at those in 40 kb bins. We also looked at chromatin interactions or loops in 10 kb at 10 kb resolution, and we looked at tab boundaries in 40 kb bins. And here on the right, it's again just showing the TADs and sort of like how the fire score across this region, in this example in chromosome 19. And so here I'm showing just basic QC of our samples. Our brain eHi-C datasets are deeply sequenced and are of high quality. Here we plotted them against all just 2 studies that had, one that had 14 tissues and 7 cell types from Schmidt et al. In 2016, and then cortical plate and germinal zone from Won et al. From Nature 2016 paper where they did eHi-C on fetal brain tissue. And so we can see that our adult and fetal samples have a lot of very usable reads for our subsequent analysis. So digging into these data sets, I'm going to talk about the properties of these eHi-C readouts. So, and the great results for biological enrichment, biological enrichment of these features. We looked at in both fetal and adult, and as a sort of negative control, we use heart ventricle. So the main takeaway is that we get a recapitulation of the key functions of source tissue, so of our human brain. For fires, our fetal samples are enriched for differentiation and neurogenesis, while our adult samples are enriched for core neuronal functions. And heart ventricle is enriched for things that you would expect, like basic myocardial function. When we looked at chromatin interactions, we get more precise connection to the tissue. So we see that gradient analysis of chromatin interactions shows that fetal samples are enriched for transcriptional regulation and for the core function of the major cell types of the brain. And chromatin interactions, gradient analysis of chromatin interactions in adult are enriched for postsynaptic density and excitatory synapse. And on the other hand, TAD boundaries are not, we don't see any enrichment, which is consistent with their sort of like cell type independent and insulative and structural feature. We then looked at the relationship between eHi-C features and the genetic architecture of schizophrenia. For this, we used the CLASS UK dataset, which is 40,000 cases and 143 hits. When we look at the fires, we see that fires seem to be the strongest predictor for the presence of a GWAS hit. And we did SNP heritability. We examined SNP heritability using partition LD score regression. And then we looked at fires, chromatin interactions, and TADs. Fire regions were significantly enriched for SNP heritability for schizophrenia. And the adult samples seem to be more enriched than fetal. And for this analysis, we use as a negative control inflammatory bowel diseases. For chromatin interactions, regions containing chromatin interactions were strongly enriched for schizophrenia heritability. And again, we observed that the adult was more enriched than fetal. And again, when we looked at TAD boundaries, we see that there's no significant enrichment, which is consistent with the null grade analysis. We used 3 different strategies to then evaluate this 143 schizophrenia GWAS loci from CLASS UK. And for all of these analyses, we focused on the adult chromatin interactions because they seem to be more enriched than the fetal. First, on the top, we followed a similar approach to Won et al. So we used FindMap to look at a subset of credible SNPs. And we followed the chromatin interactions of these credible SNPs to specific genes. The second strategy that we took was we looked at the integrating cortical EQTLs and chromatin interactions. So we looked at the conversions of a SNP that were a GWAS SNP for schizophrenia. That was also a SNP involved in a significant brain EQTL, SNP gene pair, and that had a chromatin interaction. And for this chromatin interaction, one anchor had to be at the EQTL, and the other side had to be at the actual EQTL gene. And the third strategy we took was to look at SNPs that were a GWAS SNP for schizophrenia that were located within 15 kilobases of a transcription start site of a brain expressed gene, or transcript, I should say, that were located within an open chromatin peak in adult cortex, and that were also located within an either H3K27 acetylase or H3K4 trimethylase ChIP-seq peak that were not a fire, and that there was no chromatin interaction, or they were looping to the nearby gene. And the bottom, I'm showing sort of like the three flavors that the loci can be split into. So we looked at intergenic loci, single gene loci, and then multigenic loci. So here I'm showing an example of an intergenic locus where the annotation would be original annotation would be to the nearest gene, but where we actually see a single loop going to a gene that's further away. And also this gene, you can see that there are fires in the region. They're marked by a lot of chromatin interactions within it. And importantly, you can see in the bottom, I'm showing the stranded RNA-seq data that there are no unannotated transcripts in that region. As previously observed by others, the third intron of CAC91C loops with its promoter and also within the gene. And so that's great. It's consistent with what others have previously observed. And then here I'm showing an example of a very complex multigenic locus where the eHi-C data maybe ends up providing more questions and answers into how exactly what the relationship is between the SNPs and the genes within the region. And here I'm showing a summary table for our results. So we identified 80 individual genes implicated in schizophrenia by incorporating chromatin interaction data. 9 out of those 80 are replicated from previous studies, and 71 out of the 80 are novel. And a few of these are replicated in other studies that are unpublished that are soon to be published. And I want to break it down. And so by using FindMap, we were able to identify 10 genes connected to schizophrenia GWAS SNPs. By incorporating the EQTL data and the chromatin interaction, we were able to pinpoint to 17 genes. And by additionally adding in open chromatin and histone data, we were able to identify 33 genes. And breaking it down into intergenic, single gene, and multigenic loci, we identified 9, 16, and then only 1 gene from the multigenic. So showing just how difficult it is to really analyze those regions. And so I want to thank all of our collaborators. This is truly like an effort of many because it's a very complicated procedure. The analysis is very complex. So thank you to our lab, thank you to our collaborator Yumi here at UNC, Will James Lab at Case Western, Minghu at Cleveland Clinic, and obviously the Cardiff for sharing the GWAS data. And I want to finalize with like a mystery. So I think generating functional genomics data is critical to really break down our understanding of schizophrenia and GWAS loci and all their complex disorders, but there are many unknowns. And as an example, I want to leave you with this multigenic locus, which there are many. And I think it'd be important to to really dig into this and use tissues that are relevant for the disorders to do this.
Excellent. Paola, thanks very much indeed. That was really, really good. And we've had, very minor part to play in this, but I've certainly been involved enough to know how much work goes into generating these data. And I think they're really valuable for the field, and yeah, very well done.
And it's usable for many other disorders because it's, it's controlled tissue. So I think that's the data set hopefully will be useful for many other complex disorders related to the brain.
And is the plan to share the data set once publication?
Yes. Yeah. So this is all unpublished. Yeah, we're trying to get this published. And once that happens, yes, we'll share it with the community. And we're interested in doing other types of analysis, both looking at development and other disorders. Yeah.
Okay. So we don't have any questions, but I was going to just ask one question, and this relates to what seemed like differential fire enrichment across fetal and adult schizophrenia, but for IQ it seemed to be in the reverse direction.
Yep.
Have you got any comment to make about that, or explanation possibly?
So, and I should point out that I didn't mention it, but we see a lot more chromatin interactions in fetal than adult brain. So like this is, I think, almost twice as many interactions in fetal than in adult brain. So, I mean, I don't know, maybe it means that, like, I mean, your IQ is more set from the start, like, like in a way, then I don't know if it has to do with the fact that schizophrenia is more of an adult disorder versus or not, but I think it's still, we have a lot of questions regarding why we see a difference in number of interactions, what's behind that. And we would like to try to tease apart more exactly like why do we see the specific enrichment in adult and fetal, if this is something that's common or not across other disorders.
Yeah, and is there any eHi-C data that taps intermediate developmental stages between fetal and adults?
Not yet. No, I think it'd be really interesting for like adolescent brain, etc. But for right now, this is about it because it's so expensive and pretty difficult to carry out. Yeah. Okay.
And we've had one additional question quickly from Ed Cook, who says, is there any evidence that a locus such as the one you're showing on the screen might have a set of genes or gene order more conserved in evolution? Perhaps there's a reason such regulation and the single gene in this context may not be fruitful.
Right. I think, so we looked at conservation. There's I think we can glean some information from like the TADs, fires, etc. insofar as conservation, yes. Breaking it down into genes per locus, I think a lot of these genes seem to be like very highly intolerant to mutations. So the ones that come up many times from the interactions. And I should say that for loci such as this, these are all expressed in the brain. So you can't also say, oh, well, we can ignore this gene or that one because it's not expressed in brain. So
okay. Yeah. And certainly in the original Clos-UK GWAS, we found that there was enrichment for loss of function intolerant genes, so constrained genes, not conserved, but they are related. Okay, I'm going to move on if we can, given time pressures. Thanks very much, Paola. And so now hand over to Phil Jansen, who works with Daniela Postuma. Phil, over to you if you'd like to share your screen.
Okay, thank you. Thank you for providing me the opportunity to talk here. I'm Philip, a PhD student from the Complex Trait Genetics Lab, Daniela Postuma, and happy to share our results studying insomnia in over 1 million individuals in UK Biobank and 23andMe. So the outline of my talk. So I'll first get you up to date on the genetics of insomnia, so sleep problems. Tell us something about the GWAS results that we obtained by combining UK Biobank and 23andMe. Then walk you through the GWAS follow-up analysis that we did and some conclusions that we can draw from these analyses and future directions where we would like to take this project. So insomnia disorder is defined as having problems falling or staying asleep. It's highly common in the population with prevalence estimates up to 30%. Recent evidence is showing that there's a strong negative impact on health with a higher risk of type 2 diabetes and cardiovascular disease. It's interesting to note that sleep problems are highly common in psychiatric disorders, which makes it interesting to study the genetic cross-section between these traits. The genetic factors up to now are largely undiscovered. So last year we published a GWAS paper on insomnia on the first release of the UK Biobank data in about 100,000 individuals, which implicated 3 genetic loci and 7 implicated genes, with an important role here for the MAIS1 gene on chromosome 2. But these results also showed that the large majority of the genetic loci for insomnia are yet to be discovered, and larger samples are probably going to lead to additional loci, which can be accomplished through meta-analysis. So that's what we set out to do. So the aims of this project are to further improve the insight into the genetic architecture of insomnia by increasing the sample size by 10-fold. This provides additional power to find risk loci, but also boosts the power for doing interesting follow-up analysis, which is our secondary aim, to identify associated neurobiological pathways through analyzing gene set, tissue expression, and single-cell gene expression. And a lot of this follow-up work is done through FUMA, the online annotation tool that was developed by Kyoko Watanabe, our colleague here in Amsterdam. So this is the overview of the analysis plan that we had. So first of all, we performed the GWAS in UK Biobank and meta-analyzed the results with the GWAS results from 23andMe that was performed by our collaborators at 23andMe, and performed a meta-analysis in metal to get the meta-analyzed p-values, and these are the follow-up analysis that follow from these p-values. So insomnia was measured in UK Biobank through a touchscreen questionnaire when participants visited the research center. We got data about 400,000 individuals with a prevalence around 30%. In 23andMe, the data was collected, of course, through online questionnaire in a bit larger sample, 900,000 individuals, which had a similar prevalence. And we did some work to validate these questions using an external data set from the Dutch Sleep Registry, where these questions were validated in clinical cases of insomnia disorder according to the DSM criteria, where we showed that the questions in UK Biobank had a fairly high predictive accuracy for clinical insomnia disorder, whereas the accuracy was somewhat lower in 23andMe. So these are the results of the GWAS analysis that we did. So on the left side, you see the UK Biobank and a GWAS analysis in 400,000 individuals, which led to the identification of 14 independent genomic loci, with again an important role here on the chromosome 2. On the right side, you see the 23andMe sample identifying almost 150 genomic loci. And when these two GWAS summary statistics were combined through metal, we had a sample size of 1.3 million individuals, identifying 202 genomic loci, of which basically all were novel, with an estimated SNP heritability of 7% through LD score. And as you can appreciate, in this Manhattan plot, there's genetic signal all across the human genome with several peaks. Of course, the MAIS1 that was identified earlier, strong evidence again, and with several peaks at chromosome 6, 13, and 20. So yeah, these novel loci are of course really interesting, but then we set out to map these SNP-based results to genes. And for this, we used the FUMA annotation, online annotation platform that was presented by Kyoko a few weeks ago. FUMA is able to perform 3 types of gene mapping through eQTL mapping, chromatin interaction mapping based on the eHi-C data, and of course the positional mapping. In addition to these 3 mapping strategies, we used gene-based tests in Magma, and I will show you the results now from these gene mapping efforts. So here again, the several strategies that we used to identify genes. and the right side is showing a Venn diagram of the number of genes that were linked to insomnia. So in total, we were able to identify almost 1,000 unique genes. And here you see the number of genes that were mapped by the individual methods. So on the left side, you see positional mapping, the chromatin interaction mapping, EQTL mapping. So these three are all embedded in the FUMA annotation tool. And in addition, we also perform MAGMA, identifying over 500 genes. And here you see the Manhattan plot of the gene-based association test. So every dot here is a gene with the most important or most strongly associated genes annotated here. I think 12 minutes is a bit too short to go into every individual one, but in general, we observed that many of the genes were also implicated before in neuropsychiatric traits. So this is just an overlap that we'll tell a bit more about later. So the next step was to use these gene-based p-values from MAGMA to perform gene set analysis. So for this, we used the dichotomous biological pathway gene sets from MSigDB, but also analyzed gene expression from GTEx, and also used single cell gene expression to look for single cell enrichment in specific neuronal cell types. So first of all, the tissue analysis. So for this, we use the GTEx gene expression data, which covers 53 types of tissues, including 13 brain tissue types. And these association analysis were performed in MAGMA. Here on the x-axis, you see the -log10 transform p-value of the tissue types. And we applied a pretty stringent Bonferroni threshold correcting for both the MSigDB gene sets, the tissue types, and the several single cell types. But nonetheless, 5 types of tissues passing the threshold here. And I don't think it's surprising that a lot of brain tissues pop up in this for insomnia, showing the strongest association for the basal ganglia. Where we took the first PC of the gene expression of the structures belonging to the basal ganglia, which shows the strongest association with the insomnia, but also a few specific cortical areas that pop up here, frontal cortex and anterior cingulate cortex. So this shows that both cortical areas and subcortical areas seem to be associated with insomnia, which may suggest that there might be some kind of interaction between the cortical and subcortical areas going on. So these are the tissue types, and now we're moving on to the single cell gene expression, and I think these are the most interesting results, where we collaborate with the Karolinska Institute. They collected single cell RNA sequencing from several neuronal cell types in mouse brain. These cell types were clustered in groups of cells in the level 1 analysis of which obtained 24 types of cell types, and the specific cell types which were tested independently. So here you see the results of the type 1 or the level 1 analysis with a broad classification of cell types, where the medium spinal neuron popped up, showed the strongest association. This is an important neuronal cell type in the human striatum, so in the putamen and caudate nucleus, which has connection to the other parts of the basal ganglia and to the substantia nigra, but also several cortical areas of the brain. Then moving on to the level 2 analysis, so the more specific cell types, where we found 7 cell types to be associated. Among others, the mediolateral neuroblast, 3 types of those. We also observed that the D2 receptor, so the dopamine 2 receptor, medium spinal neurons were associated, were probably driving this association. And interestingly, we also observed hypothalamic neurons, which I think is particularly interesting given the well-defined role of the hypothalamus in homeostasis and circadian rhythm regulation. And also we found pyramidal neurons in the claustrum to be associated. And I think this is also a really exciting finding because the human claustrum is somewhat of an enigmatic part of the brain. Here in the insular region of the brain, a gray matter strip. Its function is largely unknown, but we do know that it has long-range connections with several cortical areas. And transmagnetic stimulation studies have shown that this area plays an important role in sleep-wake regulation and consciousness. So I think this was really cool that we were able to go from these 1.3 million individuals to the cell types in this specific area of the brain that probably affects insomnia. So moving on to the genetic overlap with previous studies. So we performed LD score regression with previous traits, but also some other sleep-related traits that were available in the UK Biobank. And we used Mendelian Randomization using the GSMR package to say something about causality of insomnia on other traits. So first of all, the LD score regression results. Here you see that we observe strong positive associations with basically all psychiatric disorders, with strong associations with depressive symptoms and anxiety disorders. Whereas we observe negative associations with, or correlations with several cognition-related traits, but also longevity and subjective well-being, which I think confirms previous work. And most of these associations were significant due to the large sample size that we had. I will quickly skip over this. Then we performed Mendelian randomization using the GSMR R package that was published this year in Nature Communication by Su et al., which uses GWAS significant SNPs as the instrumental variable to test directional effects between traits. So instead of having correlation, this gives you an idea of which trait is causing which. So on the left side, here you see several traits that had significant genetic correlation with insomnia, and on the left side you see the effects of insomnia on these traits, where we showed that the type 2 diabetes, that insomnia had a significant effect on type 2 diabetes, BMI, coronary artery disease. But when we tested the opposite direction, there was no significant effect. So this suggests that insomnia causes these metabolic syndrome-related traits.
1 minute, Phil, please.
Sorry? 1
minute, please.
1 minute, okay. When we looked at the psychiatric traits, we saw associations in both directions. And interestingly, for schizophrenia, we found that insomnia had a causal effect or a directional effect on, or insomnia had a directional effect on schizophrenia. Whereas if we looked at the other direction, this was also not present. So I think this is an interesting finding that you can use, that you can draw from just summary statistics. So to conclude, we performed the largest genome-wide study of sleep. Importantly, we identified a lot of novel loci genes, tissue type and cell types. And I think specifically the neuronal cell types that we were able to identify through the single cell RNA sequencing were really interesting, which may provide possible targets for further experimental studies in the lab. So if you want to read more about this, we posted our work on bioRxiv beginning of this year, which also covers additional sleep-related phenotypes that I didn't have time to go into. And I would like to thank my collaborators and my colleagues from the FU, especially Daniel Postuma and our collaborators from Karolinska, who did all the work for the single cell gene expression. And I'm happy to take questions at this point. Thank you.
Brilliant. Thank you very, very much indeed. And it was good to see your colostrum. Mm-hmm. Very beautiful it was. Thanks for sharing that with us. In the interest of time, I'm going to move on. There's no questions have been submitted, but perhaps people can direct questions through the chat facility to you if they wish. But it was really interesting. It's brilliant that GWAS numbers are approaching, well, are beyond 1 million now, and I'm sure people might have questions about how you handle that amount of data. So it's my role now to hand over to Nick Clifton, who's here with me in Cardiff, and I'll get Nick to come and share the screen. Thanks, Phil.
Yep.
Right. Okay. So firstly, thank you very much indeed to the PGC, to James and to Pat for inviting me to talk today. I'm going to talk about this group of genes targeted by FMRP and the role that they have in psychiatric disorders, or at least the inferred role by the association through genetic variants. So in 2001, Darnell and colleagues published this work, and they identified a set of genes that were bound by FMRP. Now FMRP stands for Fragile X mental retardation protein, and it's coded by this FMR1 gene, mutations in which are known to cause Fragile X syndrome, and the role of FMRP is in regulation of the translation of these mRNA species into proteins. And Darnell and others proposed or discovered that the FMRP has hundreds or perhaps even thousands of targets. They performed quite a stringent cutoff and identified a set of around 840 genes targeted by FMRP, but that was a fairly stringent cutoff, and there are likely many more than that. And then since then, that gene set's received a lot of attention, and also there was this paper in particular which pointed out the controversy around these targets. So it was recognized correctly that the FMRP targets tend to label genes that are very long and very highly expressed in the brain, and so it was important to identify whether their role in their implication in psychiatric disorders goes beyond these potential covariates that could actually be acting, implying that FMRP could be a proxy for these other characteristics. So it's important to know whether there was a relationship between the probability of a gene being an FMRP target and the gene's association with psychiatric disorders. So what I did is went back to the Darnell publication, and you find in the supplementary information all of the genes in the mouse genome. These FMRP targets were identified in mouse cortical tissue, and they ranked them by the probability of being an FMRP target. Okay, and so you find genes at the top of the list that are most likely to be targeted. And what I did is group these genes into 41 bins of 400 genes. So bin 1 would contain gene 1 to 400, and bin 2 would be 401 to 800, etc., etc. Okay, so that we can dissect the association of FMRP targets with psychiatric disorders by looking across their probability. So what we can see here in this example, using PGC, CLOS UK meta-analysis of common variant summary statistics, that there does appear to be this association between genes which are more likely to be FMRP targets, and up on the y-axis here we have the strength of their genetic association with schizophrenia through common variation. And you can see here that it's indeed the top 2 bins that are most strongly associated. And this dotted line represents a threshold after correcting for all of these bins. And what's particularly powerful about this magma analysis that we use to do it is that it corrects for gene size and SNP density. So that the effect that you see here is above and beyond that would be mediated by these covariates here. Okay, so what I've looked at is the association of these bins across different types of variant. It's one of the most interesting things about the FMRP targets gene set is that it has been associated across different types of variant. So here you can see an analysis performed by Elliot Rees of the rare variant association that, again, just the top most bins. So those bins most likely to be FMRP targets are those that are strongly enriched for association with schizophrenia through rare variation, and also through de novo rare variants as identified through exome sequencing as well. You can see just the topmost bin here is enriched. Conversely, through CNVs, copy number variants, we didn't see the same association, and there's various reasons why that might be. Copy number, the association of FMRP targets through copy number variation has been identified as quite small in the past anyway, and it's perhaps an artifact of it being quite difficult to identify the causal genes from within copy number variants. So what we then did is performed some gene ontology and enrichment analysis on the traditional Darnell FMRP target gene set. Now this, we're not the first people to do this, but it was a necessary step for the subsequent analyses that we performed. And when you perform this analysis, we found 169 Go terms were enriched in FMRP targets. And we used a refinement procedure to identify 21 terms for which they had a independent enrichment in this set. And feel free to ask me about this, this procedure if you're interested for refining gene terms. Okay, so these are the 21 terms that we picked out. And fairly unsurprisingly, many of these are related to synapse function or to development of synapses. Remember that these FMRP targets were identified within mouse cortex, and also one of the roles of FMRP is in the translation of mRNAs to proteins, and it's thought that a lot of that is performed at the synapse locally as well. So, but what this allowed us to do is to group these together into a further gene set that I've called synapse-related genes, about 2,000 genes that allow us to further refine the signal that we see from FMRP targets. And I'm going to talk about this in the context of schizophrenia here. So these synapse-related genes themselves, when you analyze them for enrichment for association with schizophrenia through common variation, you see that they are indeed enriched, and as we know, these FMRP targets. Okay, but what we're interested in is how does the signal divide between those FMRP targets that fall within this functionally defined synaptic groups and those that don't overlap with these terms. So when we perform this magma analysis, just like before, interestingly, we find that that enormous proportion of the signal from FMRP targets exists within this overlap. So the p-value here is very small, and the p-value from those FMRP targets that don't overlap with synapse-related genes is much smaller. And when you compare these betas, you indeed find that they are significantly different. And further than that, we can perform a conditional analysis, which is asking whether these 367 genes hold a further enrichment than that you get from synaptic genes and FMRP targets. So this is particularly interesting because it shows that not only are these two groups of genes important in psychiatric disease, in schizophrenia, but also it's those genes that are both synaptic and targeted by FMRP that seem to harbor most of the enrichment. So we then wanted to look at the association of FMRP targets across disorders via common variation. So I've shown you this plot already, which is schizophrenia common variants. But it's interesting to see that you see the same relationship with bipolar disorder as well. This is using PGC data in a recent bioarchive. study. So just the topmost bin, FMRP target bin is enriched there. And also for major depression, just the topmost bin. And as a negative control here, we have Alzheimer's disease common variation from IGAP, where we don't see that same relationship. So interestingly, when you look at the division of the signal between between those FMRP targets that overlap with the synapse-related genes and those that don't, in major depression, you see this division much like schizophrenia, where most of the signal exists within the overlap. However, for bipolar disorder, the division is spread much more equally. In fact, perhaps slightly more in those that don't overlap here. So this perhaps is a way where disorders can be dissociated by the genetic signal here, and we're writing up this work at the moment, and hope to look further into what these genes are that are enriched in bipolar disorder. So just to give you a hint at what these genes may be, when you perform a gene ontology enrichment analysis of this segment here, the terms that come up tend to relate to chromatin modification or regulation of transcription. So what we're going on to do is look at the role of these types of genes in bipolar disorder as implicated through genetic variation. So just finally to thank these people in particular, specifically Andrew Pocklington and Elliot Rees have contributed a lot to this work, and it's my funders as well. Thank you very much.
Thanks, Nick. Brilliant. So we'll just stop sharing and then look if there are any questions. Okay. Anyone got any questions for Nick? Fine. Well, I'm aware we've just about made the 4 o'clock. I think we're 1 minute, 2 minutes over, but I'd like to thank each of the presenters today. I think this format, hopefully people have felt, has worked. It's quite intense. There's a lot of data, but actually I think it shows us that the PGC moving forward is going to be in very good hands. And again, thanks to all the speakers for agreeing to share their work with the community today. Thanks very much. We'll see you at the next Will Lab meeting. All the best. Cheerio. Thank you.